I now have the great honor and privilege to essentially introduce today's three plenary speakers that will start at 4 o'clock, which is about a minute or so from now. And they'll present their thoughts on addressing the grand challenges for engineering from their perspectives in, of course, research, government, and academia, all critical components for solving the grand challenges in the centuries to come. It really gives me enormous pleasure to introduce Robert S. Langer, the David H. Koch Institute Professor at Massachusetts Institute of Technology, and Dawn C. Myricks, Deputy Director of Science and Technology at the CIA, and Thomas Katsouleos, who's now Executive Vice President and Provost at the University of Virginia. Speaking for us today first is Dr. Langer. He has been awarded almost every award that's possible to win, including the NAE Draper Prize and the Queen Elizabeth Prize for Engineering. He's written over 1,300 articles, has over 1,080 patents worldwide, and is the most cited engineer in history. Otherwise, hasn't done very much. Today, he will talk about the grand challenges, the particular grand challenge, engineer better medicines. Bob, the floor is yours.
Thanks so much, Phil. Thank you. Well, thank you so much, and congratulations to all the new members. It's just wonderful to see the NAE doing so well and having such terrific people in it. You know, so I'm going to go over my perspective, at least with 3 examples, on grand challenge number 3, which, as was mentioned, is engineering better medicines. The first one, if we could put up the first slide that I was going to go over, and actually there are many more than these 3, but was, can we create new and better biomaterials? And to illustrate that, I think what I wanted to do is start out and tell you at least my perspective of where we stand with biomaterials today. And interestingly enough, for me, when I started my career, the very first job I had after I got my doctorate from MIT was working in a hospital. And I did started doing a lot with materials. But one of the things that I noticed was, how did materials find their way into medicine? I thought, actually, and I was very naive, that it must be engineers or material scientists or chemists that were the driving force for this. But when I looked at this, almost every time I saw the driving force for materials in medicine were medical doctors. And what they did, because they urgently wanted to solve a medical problem, is they'd go to their house and they'd find some object in their house that kind of resembled the organ or tissue they wanted to fix. And let me exemplify that. But it's, you know, I realize this slide may look funny, but it's also 100% true. So if you look at it, I'll pick the first example, 1967. In the case of the artificial heart, some of the clinicians at NIH and elsewhere, they wanted something with a good flex life. And they asked, what material has a good flex life? And they said, a lady's girdle. So that's what they made the artificial heart out of in 1967, was the material in a lady's girdle. That, by the way, is still what it's made out of today. And because once you start down that path from like an FDA regulatory standpoint, it's not so easy to change. But as many of you may know, that one of the problems with the artificial heart sometimes has been when blood hits the surface of the artificial heart, it can form a clot. The clot can go to the patient's brain. They could get a stroke and they die. But if you think about it, something that's designed to be a lady's girdle, you know, it may not be the optimal blood contacting material. And this problem literally pervades almost all of medicine. Dialysis tubing, the sausage casing, vascular graft, that's artificial blood vessel, was a surgeon going to a clothes store in Texas, finding what he could sew well with, and picked Dacron clothing. And breast implants, one was a lubricant, the other actually a mattress stuffing. You can probably think of the logic. And I remember being a young chemical engineer thinking about this, and you know, one of the things you learn in chemical engineering is design. And I thought, well, why can't we use chemical engineering principles to design new materials? And we picked an example. When we started, the only material that was approved by FDA that was synthetic and degradable were these polyesters that showed bulk erosion. And that's okay for delivering some drugs, but you can see they degrade throughout, and that means when they degrade, that could lead to bursts of drug release And let's say when this collapses, if you had a drug like insulin or an anti-cancer drug, you might dump out a lot at once. So what we and others would say was that what you really want from an engineering standpoint is surface erosion, kind of like the way a bar of soap dissolves. And could we do that? And what we did, and I won't go through all of this, but we went through a detailed engineering analysis, sort of step by step, and came up with the idea that a particular family of polymers, polyanhydrides, I won't show many chemical formulas, I promise, could, if they had the right building blocks, could do this. And we also proposed that once we had created these, that maybe they could even lead to some new medical treatments. And here I began working very closely with some neurosurgeons like Henry Brem, who were at Johns Hopkins. Henry today is chief of neurosurgery. He's a member of the National Academy of Medicine. And one of the things that they wondered about was, could we come up with a new treatment for brain cancer? And in particular, the idea that we came up with was, before this, people always gave chemotherapy throughout the whole body. And it causes terrible side effects to liver, kidney, and spleen. We wondered, with these new materials that could deliver drugs very precisely, and we published many papers on this, could we allow the neurosurgeons like Dr. Brem to do the following? They're going to operate on the patient anyhow to take as much of the tumor as they can out. But before they close the patient up, could they line the surgical cavity with polymers containing this drug, BCNU, which was the main drug at the time that people were using and still is widely used. BCNU, the drug, normally has a half-life in the body of just 12 minutes. But what we thought is, if we put it in a polymer, the polymer could protect it from degradation. We could actually make these polymers last any length of time. So we could actually then make a little wafer that could be put in the brain, I'll show you this, that could last for a month, locally deliver the drug to the tumor, and get high concentrations in the brain where you wanted it to, and low concentrations in the rest of the body where it causes harm. Now, one of the things I think, and this is going to be true for probably almost everyone in this room who's been inducted, but I'll just share my personal experiences, especially for the academics. You know, if you come up with an idea like a new family of polymers, you know, maybe new therapies, you know, you always have to raise money for this. The way we would generally raise money is we would write grants to the National Institutes of Health and other organizations, and then they'd have people review it and tell what they think. And we didn't do very well on that. I actually saved a lot of my reviews. So when we first proposed this, people said, you know, that wouldn't work. So actually in 1981, when we first wrote this, the chemists actually said you'd never be able to synthesize these polymers. But I had a very good graduate student at the time named Howie Rosen. Howie is actually in this audience today. He's a member of the NAE. He later became president of the Alza Corporation, a very, very successful drug delivery company acquired by Johnson & Johnson for $12 billion. And he synthesized it. But that wasn't enough. We sent the grant back, and the reviewers looked at it and they said, well, that's nice, but you know, you have these anhydride bonds. The chemist said, they'll react with whatever you put in. But I had another postdoc, Cam Leung. He's now chaired professor at Columbia. He's also a member of the National Academy of Engineering, and he showed that that didn't happen. We sent it back again, and the reviewers said, well, but these polymers are low molecular weight. They'll break in the body. But I had another couple postdocs, Avi Dome, who later became head of medicinal chemistry at Hebrew University, and Edith Mathiowicz, who's full professor at Brown University and now a member of the National Academy of Inventors. And they showed that you could make very high molecular weight, strong polymers. That didn't happen. So then people said, well, okay, that's nice, but new materials are certainly going to be toxic. I had another graduate student, also in this audience today, Cato Lorenzen. Cato is a member of both the National Academy of Engineering and the National Academy of Medicine, and he actually showed they weren't toxic at all. He also became Dean of Medicine at the University of Connecticut, among other things. Sent it back again, and the reviewer said, well, that's good, but you know, the drug won't diffuse far enough. But I had another graduate student, Mark Saltzman. Mark is now head of bioengineering at Yale, and has been elected to the National Academy of Medicine. Anyhow, this kept going on and on. until 1996, when the FDA approved this treatment for brain cancer. It was actually the first time in over 30 years in over 20 years they approved a new treatment for brain cancer. The first time they ever approved this idea of local chemotherapy. You can probably tell from the way I'm speaking that I'm very proud of how well all our graduate students and postdocs at MIT did, many becoming members of National Academy of Engineering, and all, I think, having remarkably successful careers. The reviewers, they haven't done that well. Now, I would like to show you what this operation looks like, but if anybody's squeamish and doesn't like the sight of blood, you shouldn't look. But just here, this is what it looks like, a little wafer going into the brain. I should say it's very hard when I give a talk to get good advice, but my wife, one time when I was lecturing at MIT to a group of chemical engineers, went to one of my talks, and at the end of it, I said to her, well, what did you think? And she said, well, Bob, she said, the talk was okay. It's actually very high praise. But she said, I don't know if you realize, but you had that bloody slide on for over 10 minutes, and all the poor engineers were turning green. So ever after that, I did what I did today. I gave a warning. I showed it very quickly. But I just want to tell you the sequel to that story was that 2 years later, I got asked to give this dinner speech. It was actually to a group of neurosurgeons. And I told them the same story. And at the end of the talk, quite a few of the neurosurgeons came up to me, and they said, that bloody slide you showed? I said, yes. They said, that was fine. You could have left that on as long as you wanted. They said, but that chemical formula, to leave that on for a neurosurgeon. Anyhow, so I kind of adjust what I do. If I lecture at the National Academy of Medicine, it's a little bit different talk. At any rate, this is just some of the clinical data. Some of the clinical data. What you see is, it's not a cure. There is no cure for this, but you get you do get a prolongation of life, factor about 5-fold improvement in survival, and fewer side effects. This now has been approved, been used the last 19 years in over 30 countries. But more importantly, I think it began to set a paradigm of local delivery, and that's now used in all kinds of new ways. Maybe the most predominant way where it's made an impact actually is in drug-eluting stents. It's really the same idea. You have a stent. If somebody has heart disease today, they often put these stents in to keep the blood vessels open, but about 60% of the time they'll close because of smooth muscle cell proliferation. Now what's done, and some of this actually was done by Elazar Edelman, another one of my former students who's also a member of NAE, and companies like Boston Scientific, is to coat these stents, which are like Chinese finger puzzles, with a polymer, now a different cancer drug, Taxol. And that's been remarkable. I mean, about 1 million patients each year use them. And there are many other challenges in drug delivery. Nanotechnology, where you could deliver siRNA or new gene editing techniques, messenger RNA, non-invasive delivery of drugs, which Samir Mitragotri, another one of my former students who's just been elected to the NAE today and at UCSB, has been pioneering, and many other challenges as well. But I wanted to now go and tell you one that I'm also excited about for the future. Could we even make these smart? And as I'll show you, we can maybe even do things to help the third world as well. A number of years ago, I was watching a television show on PBS, and what they were talking about was, could they make microchips for computers? And I thought when I watched this, wouldn't this be a great way to make a drug delivery system? Now again, if you spent 30 or 40 years of your life working on drug delivery systems, you might think that about any TV show. But I had this idea, and I contacted Michael Sema, one of my colleagues across the hall. He's also a member of NAE now. And we came up with this idea. We got a young graduate student, John Santini, who was originally a junior at University of Michigan. And we made these chips. This is just a picture. This is like a cutaway of the chip. I'll show you more about it in a few minutes. But the idea is that you could have different wells. The wells could contain the same drug at different doses, or it could contain many drugs. It could literally, if the drugs are potent enough, you could have a pharmacy on a chip. And we have covers, which could be metals like gold or platinum alloys, and they would cover it. But as I'll show you, we can pop these covers off almost by remote control. So we can make these delivery systems deliver the drug when we want to. Let me just show you a little bit about what we did. So this is just an example of a well that we had, and we might put up to 100 wells on a chip. And this is before activation. But once we turn on, by remote control, an electric field, the cover comes off. And like I say, we could have a tiny chip that's no bigger than like a dime, and we could have probably up to 400 on a chip. And so you could have 400 doses. There's all kinds of ways. And by the way, over the years in our lab, we've made like chips that can be injectable, like through trocars that are cylindrical. We've made chips that can be short and flat. I mean, pretty much any size and shape. We've made them that are totally degradable as well. We published in Nature in 1999 the first example of this, where we put different doses of drug in different wells and triggered release at different times. And here's an example of that. This is just in test tubes. To more do like the pharmacy on the chip, we put multiple drugs in. These are just model drugs, and triggered release at different times. But then what we did is we started a little company called Microchips. Now it's called Microchips Biotech. In fact, the CEO of that company is Cheryl Blanchard, who's here in the audience today, who has also just been elected to the National Academy of Engineering. I don't want you to think everybody in this room worked with me, but at any rate, but there are a few. At any rate, so Cheryl's done an incredible job, and I'll get to some of where we are today in a minute. But basically, over the years, we moved from in vitro studies to small animal studies, to large animal studies, to actually human beings. And what I'm going to talk about might almost sound like space-age medicine, but it's really being done. The first clinical trial was done actually in Denmark. Chips were put in the human body, and you can actually communicate with them over special radio frequency called the Medical Implant Communication Service Band. It's approved by both the FCC and the FDA. And again, I think there's bigger problems we're worried about than tampering, but nonetheless, you can have the patient or doctor enter a special computer code to administer, change the dose if that needs to be done. And there's actually a bidirectional communications link between the chip, which is in the body, and the receiver, which could be something like this, or it could be a cell phone, or any kind of thing. And it tells you all kinds of information that you normally don't necessarily know, like, did you take the drug? Which I have to admit, as I've gotten older, I think I might need more and more. And other things like battery life and so forth. So let me just tell you a little bit about the first clinical trial. It was 8 patients, and we wanted to pick an example that we felt could really, A, give us a tough test, and B, make a clinical impact. And what we chose was to try to treat osteoporosis. So let me just tell you a little bit about that. So osteoporosis is particularly prevalent among older women, and one of the ways that that's treated is to give an injection of a large peptide, a parathyroid hormone, 39 amino acids, once a day. You don't want to give it continuously because that will actually cause bone resorption. You want a pulse once a day. The problem is if you have to inject it once a day, the dropout rate, patient compliance, which is a huge issue, is 75%. Most people don't do it. I mean, who wants to inject yourself once a day? So what was done is to do this small office procedure in the doctor's office to do the implant. It takes just a few minutes. And then what we see, and I'll show you this in a second, is you get the same pharmacokinetics, that means the drug kinetics in the body, with actually less variability than injections, which I don't think matters in this case, but may matter in other cases. And then there are 3 measures, for example, of how you look at whether you're treating the disease, calcium, and then these collagen fragments as daily injections. And here's the data. This is days like 60, 68, 76, and 84, superimposes parathyroid hormone concentration. This is an early stage chip, which has the chip itself. It has microelectronics. It has a battery. It has a computer program. And there's even an antenna built right into it that you can communicate with. There is minor fibrous encapsulation, but not much, and certainly does not prevent the drug from getting out and doesn't change over time. So this is one of the areas, and in fact, now we're looking at various other clinical indications with this as well. But what's nice about a brand new technology like this is sometimes you can come up with areas that you don't even think about because you that's not where your head's at. But other people come in and visit and they think, well, maybe you could use it for this. So I'll tell you a story. A couple of years ago, I had a visitor to my lab. And this person was not a chemical engineer. He's actually a very successful software entrepreneur. He also is a member of the NAE. I'll just show you. Here he is. Here I am. And he said, well, you know, he and his wife, in fact, we're working on about 7 projects with him, said he and his wife have this foundation. If people don't recognize him, it's Bill Gates. They have this foundation, and one of the things that they were interested in, certainly there are lots of grand challenges here, is there are women in the third world that get abortions all the time. It's terrible. Lots of women die. And one of the big issues is birth control. Could they actually have a birth control device that could be inserted and last for like 17 years, perhaps, and that the woman, if anytime she changed her mind, could turn it on and off whenever she wanted? Now, you cannot do that with any existing device. First, none last more than 5 years. Secondly, none of them give you the ability to turn on and off, and they're hard to remove in many cases, particularly in the third world. But with the microchip, we thought we could do that. And we're doing that. They've actually funded Microchips Biotech. This is actually right from the Gates website, to develop a personal fertility control system with emphasis for use by women living in developing world countries as a means to effectively plan their family. So far, invested $13 million. And we hope that there are many other things that this can be used for too. This is the implant today. Like I say, it's a quarter with 100 wells. And this is what it looks like. It's actually a very interesting combination of electrical engineering, chemical engineering, and material science, but that we hope will be able to address this challenge. And the very last challenge that I want to address is, could we someday create new tissues and organs? Starting in 1983, Jay Vacanti and I, Jay's a surgeon at Mass General, came up with this idea. He's a pediatric surgeon. He would try to do liver transplants in patients, and he could just see people dying in front of his eyes. And he asked me, could we help? Could we come up with a better way to treat these patients? And the idea, this was a paper we published in Science that's been cited 5,400 times now. But basically the idea is that you could take any cell type, including stem cells, but we wrote this before people used them. But this is bone, cartilage, liver, intestinal, urothelial. Could we do this? Could we put them on a polymer scaffold? And the idea is, by putting them on a polymer scaffold, if you have the right chemistry, the cells will stick. And not only that, the cells are smart. And other groups, for example, at Berkeley, had shown that you could take, for example, mammary epithelial cells, and if you put them close enough together, they're smart enough they could actually form acini and make milk. So our thinking was that maybe we could make various tissues and organs this way. It also is key the way you grow them. There's a lot of engineering in growing the right types of bioreactors, and then you could put them in the body. Just to show you a few pictures. This is an early scaffold that we made, where you have fibers, and in this case, liver cells. In fact, two members of the National Academy of Engineering, Tony Mikos, who's now at Rice, David Mooney, who's now at Harvard, both students of mine, really did a lot of pioneering work in the 90s on this to help create the foundation for these scaffolds. And then you could even go further. Let's say, and this is all speculation, but let's say 30 years from now, somebody doesn't like their nose, and they go to a plastic surgeon, they say, I'd like a new nose. So to that end, Prasad Shastri, another one of my postdocs, he now runs an institute in Germany, said, well, why can't we use CAD-CAM techniques like computer-aided design? Maybe what we could do is have a computer screen with 40 noses. And by the way, this isn't limited to noses, but this is. So what he did is so here's the idea of a nose. This is 98% porous with an FDA-approved biodegradable polymer. Now this is a regular nose, but let's say somebody wanted an upturned nose. Well, that wouldn't be so hard with polymers. Cartilage is really tough to cut. Cartilage is what your nose is made out of. But with a polymer, if we wanted to make a little bit less, you just take a little bit of this off. Let's say somebody wanted an upturned nose. I mean, they probably wouldn't. I'm sorry, a hook nose. They probably wouldn't. But like upturned nose, you could do this. Hook nose, you could do this. And you could make them with the CAD-CAM approach, followed by a technique like 3D printing or other fabrication techniques. And that's what he did. So here we have the nose with the cartilage cells on it. And then you could put it in the patient. And I thought I would just close with two examples of where these are being even used at different levels today. One is actually approved. We've had a number of companies over the years license our technology. An early one was Advanced Tissue Sciences, and they, through various evolutions, actually made artificial skin. And let me just show you an example of that. Here's a 2-year-old boy who's very badly burned. Here's the product, which is a polymer scaffold with neonatal skin fibroblasts on it. But now you put it in the child. This is at the time of injury. Let's come back 3 weeks later, 6 months later. These are now approved by the FDA for patients who are diabetic and who have diabetic skin ulcers and burn victims. Now I'm going to show you the last example, and this is still early, but it's exciting and I think representative of the grand challenges. And what we did is, Erin Levick, another one of my graduate students, she's now a professor at Case Western Reserve, she was in the material science department. And she said, well, could I make a scaffold? People, have spinal cord injury, right? They're paralyzed. Could she make a scaffold that, using material science and engineering techniques, could mimic the gray and white matter of a spinal cord? So she made a tiny little system that you could put in, and we had the outer part be able to attract cells, including stem cells, and the inner part be porous. And basically, we did a huge study, Erin with neurosurgeons like Ted Tang, and we had stem cell specialists like Evan Snyder. So we did 50 animals, and 12 had a polymer scaffold by itself, 12 had nothing, 12 just had what we call neuronal stem cells, and 13 had the combination of the cells, neuronal stem cells, and the polymer. Let me just show you typical control. So here's the diabetic rat. Here's the paralyzed rat. The two features to notice are the paws are splayed awkwardly, and he's not able to support his own weight. There's actually a BBB scoring system. He gets a 5. The maximum is 20. And this is pretty typical of what you get by cells by themselves or a sham operation. And we followed them for 400 days. This is at 100 days. If you put the polymer scaffold in by itself, it actually does better. It gets rid of some scar tissue, and you don't get so much cyst formation. But the best results, maybe not surprisingly, were the combination of the polymer scaffold and the neuronal stem cells. And when you put that in, and this is the exact same experiment, here's the mean of the treated group. And it's not a cure by any means, but it's doing much better. Even though this guy's kind of fat, he is able to bear his own weight. And notice the paws are splayed in a much more normal fashion. And he gets a score of 14 out of 20, even though he's a bit clumsy, but he's doing okay. There are the paws. So then, again, another company, In Vivo Therapeutics, licensed this, what they did is repeat these experiments now on non-human primates with some other very good neurosurgeons, Eric Woodard and John Slotkin. And this is the primates now with this stem cells and the scaffolds, but first the control. And this is now on a treadmill 12 weeks after the start of the experiment. So this is pretty typical of the control. He's having trouble moving this leg. In a second, we're going to see this one start up. So then what we did is, based on this, the company went to the FDA to ask the FDA would they allow them to start clinical trials. Now, they could have tried to do it with the stem cells. They thought it was a lower bar to just use the scaffold by itself, which showed some activity. So they applied to do human clinical trials, and the FDA granted them that approval. They then got what's called IRB approval, Institutional Review Board approval, at 14 hospitals around the United States. And this is actually, some of this it's been kind of remarkable. But I just thought I'd give you a couple highlights and show you a video. In today's world, what happens is when patients are treated like this, they actually make videos and put them on YouTube and things like that. So the first patient was a 25-year-old young man in Arizona, and he got in a terrible accident, was paralyzed. They put the scaffold in him, and I'll just show you him just a couple months later. This is a video. My last slide. The key things are that originally he was completely paralyzed. It's a safety trial initially with the FDA, no adverse events. He had active movement of his hip flexors, palpable contractions of knee extensors. He had no bowel function initially. He's totally regained it. No bladder function initially. That's improved. So here he is. This is his sister.
That is amazing.
So the second patient was a 22-year-old woman in South Carolina, also very badly paralyzed, in the ICU for nearly a month. So she got the treatment. Interestingly, like I say, this probably wouldn't happen if we weren't in 2015, but she knew she was the second patient. He was the first patient, so I guess she emailed him or Facebooked him, and they started communicating, and he gave her advice. She also started making quite a good recovery. But then they actually wanted to meet each other, and they did, and they actually fell in love. So it's This actually has been, actually they had in June a 6-minute episode of this on the Today Show. And, you know, mostly on the falling in love, not the treatment. But at any rate, I always say, like, tissue engineering can actually lead not only to hopefully scientific advances, but other things as well. And she is making quite a good recovery. A third patient, also in South Carolina, apparently has made even a better recovery still. There was just an article in Bloomberg, I guess earlier this week on that, and the fourth and fifth patients were just treated within the last month, both in California. And that is the goal, is, like I say, largely a safety trial, but you can't help but get some efficacy data. And apparently, according to the clinicians, even though it's early and you don't want to overthink what will happen with just 5 patients, the chances of the first 3 doing what they did, there's less than a 1 in 200 chance that could happen. So obviously, a lot more needs to be done. And I just think this is the tip of the iceberg, because this doesn't have stem cells in it. It doesn't have neurotrophic factors in it. And really, both the skin and this are examples of grand challenges. My hope is, as we go forward, and we and others in this room are working on this, is could you use tissue engineering to create new hearts? Someday, new pancreases, pretty much any tissue or organ in the body. And there are no drugs for these things. But these are areas where engineers can and have made enormous contributions, and I expect will continue to. It's been an honor for me to present to you today, and again, I want to congratulate all the new members. Thank you so much for having me.
Well, thank you very much, Bob, for a really great presentation and a tremendous introduction to our membership to Engineering Better Medicines, and really opening the door to many people's view of what the possibilities are going forward. For a few more PhDs out of your program, they'll be all over the place. We'll be able to fund all our medical schools and universities. So that was really terrific.